So when we are exposed to a foreign antigen, our B cells will be selected based upon which one of them will have the right antibody, and then they will undergo cell division. And so that is the idea of producing many B cells of the right type. And that is what is referred to as clonal selection. So clonal just means undergoing mitosis, undergoing cell division, where you produce many B cells of the right type that are going to be able to fight off the specific antigen that your body has been exposed to. So we have in clonal selection, B cells that are specific. Specific. They undergo cell division and produce plasma cells and memory B cells. So plasma cells are like antibody factories. And they can actually just pump up and produce these antibodies that are then secreted into, for example, the blood. The memory B cells are long lasting. So these are long lasting. So they are what give us our immunological memory. So when we look at this process, let's look at a diagram. Remember that our B cells are produced randomly. We are infected with a microorganism, and on its surface, it has specific proteins that are antigens. These antigens only bind to specific B cells, so the specific B cells that have antibody receptors on their surface. So here we see the activated B cell. This B cell is then going to undergo cell division, and it produces these plasma cells that secrete antibodies in abundance. And these antibodies can bind to the antigen and then neutralize the microbe, or they can prevent the microbe from being able to do damage to the body. And then we have these long-lived memory B cells, which stay in our body so that if we are exposed to the antigen again, we will be able to produce antibodies much more quickly than we would have been able to do before. You'll also notice that we have the helper T cell here. So the helper T cell gets a signal from the B cells to activate the cell-mediated immunity component. And that cell-mediated immunity would aid in helping us fight off the infection.